This is the second video of the Cell Death Pathways lecture. In this lecture, we will talk about apoptosis. So, apoptosis is a form of cell death in which a programmed sequence of events leads to the elimination of cells without releasing harmful substances into the surrounding area. One of the biggest differences between apoptosis and necrosis, as we saw in the previous video, is that this process cannot be stopped once it has begun. Apoptosis plays a crucial role in developing and maintaining the health of the body by eliminating old cells, unnecessary cells, and unhealthy cells. But it also contributes to pathological processes that are involved in neurodegeneration. Apoptosis can be initiated through one of two different pathways, and we will see this in more detail in the next few slides. But there are two basic types of apoptosis. There is the intrinsic pathway, in which the cell kills itself because it senses cell stress, or the extrinsic pathway, when the cells kill itself because of signals from other cells. So there are three key molecules that play an important role in apoptosis. One of those key molecules are the BCL2 family proteins. The other is the APAF1 or apoptotic protease activating factor 1. And the third element are caspases. And we'll see in more detail information of each of these components. So in the case of the BCL2 family members in their role in neuronal death, there are two types of BCL2 proteins. There is one group, or group number one, which belongs to anti-apoptotic, or it's related to anti-apoptotic functions. Those are the BCL2 and the BCLXL proteins. And these proteins share different regulatory domains, different BCL2 homology domains, and similar regulatory processes. However, there is other group of BCL2 proteins that belong to those with pro-apoptotic functions. And those are such as BAX, BAD, BEAD, PUMA, or BIG. Those are all proteins that have pro-apoptotic functions, and they share some of the BCL homology domains. The regulatory processes are different, and they are mainly regulated by tumor suppressor protein, or TP53. So they're similar in like structural wise, but they have completely different functions, and they're regulated by different factors. So the BCL2 family proteins are, as we just said, the regulators of apoptosis, but they also have other functions. This family of interacting partners includes inhibitors and inducers of cell death. Together, they regulate and mediate the processes by which mitochondria contribute to cell death, known as the intrinsic apoptosis pathway, and we will see this in more detail in the next slides. This pathway is required for normal embryonic development and even for preventing cancer. However, before apoptosis is induced, BCL2 proteins have critical roles in normal cell physiology related to neuronal activity, autophagy, calcium handling, mitochondrial dynamics and energetics, and other processes of normal healthy cells. And the relative importance of these physiological functions compared to their apoptosis functions in an overall organism physiology is really difficult to decipher. But apoptotic and non-canonical functions of these proteins are or must be intertwined to link cell growth to cell death. So the other component or key component of the apoptotic pathway is the APAF or apoptotic protease activating factor 1, which is a factor that transmits an apoptotic signal from the mitochondria to caspases. So APAF1 forms a hetero-oligomeric complex with cytochrome C, which is released from the mitochondria, and pro-caspase 9, called an apoptosome. And caspase 9 now becomes activated by the APAF1's activity. And this signals to other caspases to execute the apoptotic pathway. And this is the third and perhaps the most important component in the apoptotic pathway. Caspases are essential proteins in apoptosis and are really biomarkers for the apoptotic pathway. So caspases are cysteine aspartate proteases, which means that they can cleave in proteins possessing motifs containing an aspartic acid. These proteins are produced as inactive zymogens, which are catalytically inactive proenzymes. And we can see in this cartoon how a caspase is organized. It has a pro domain and a large and a small subunit. And caspases possess a points of cleavage between these different domains, and cleavage is activated upon the initiation of the apoptotic pathway. So what this cartoon is showing is that this long peptide corresponds to the inactive procaspase or the precursor. And then when it gets cleaved, then the different domains, the pro domain, the large, and the small subunits are truncated. So the large and the small subunits then can associate to provide the active site of the enzyme. And in this picture or in this Western blot, you can see how the procaspase 3 has a higher size. So this is the inactive procaspase. And then upon stress stimulus, then caspase gets cleaved, and then you can see these very two characteristic bands of the cleaved caspase, which corresponds to the large and the small subunit. So activation of caspase 3 can be used as a biomarker for apoptosis. So the different caspases can be subclassified into three different types. There are the initiator caspases that include caspases 2, 8, 9, and 10. And probably the most important ones or the most studied ones are caspases 8 and 9. Then this type of caspases are actually activated by homodimerization. Then we have the executioner caspases, in which we can find caspases 3, 6, and 7, and perhaps the most studied ones and the most important one is caspase 3. These caspases are activated by the initiator caspases, and these ones require cleavage and association of the large and small subunit. So the initiator, as the name says, they start the apoptotic process, and they activate other type of executioner caspases that are in charge of finalizing or executing the apoptotic pathway. And then there is a third group of caspases that are related to inflammation, and activation of those don't necessarily mean that there is apoptosis happening. Some of those well-known inflammatory caspases are caspase 1 and 11, and there are also markers for cell death pathways such as apoptosis and others, but we're not going to talk about those in class. So in this diagram, we can now start putting together all the different pieces and the different components that we've been talking about in the previous slides. And this is how the apoptotic pathway can be kind of like divided. So we mentioned earlier at the beginning of the video that there is extrinsic apoptosis and intrinsic apoptosis. So, well, what do differentiate the two type of apoptotic pathways? Well, the extrinsic apoptotic pathways are activated through ligand death receptors. And this is a very specific family of receptors that can sense molecules that are triggering apoptosis. Those receptors are the membrane that are associated to caspases. Procaspase 8 is actually a caspase associated to the extrinsic apoptosis. And once the receptor binds the ligand, then it causes procaspase 8 to dimerize and activate bead proteins. Those bead proteins go into the mitochondria and start a mitochondrial disruption process that includes membrane permeabilization, the pore disruption, etc. Then on the other side, we have the intrinsic apoptosis. This type of apoptosis senses intracellular changes or intracellular stresses. One of the well-known activators for intrinsic apoptosis is DNA damage that responds activating P53 or tumor suppressor P53. And then as we saw earlier, P53 is responsible for activating some of the pro-apoptotic BCL2 family members such as BAX, and also inhibit BCL2 and XL isoforms, which are the anti-apoptotic ones. So there is a complex mechanism between BAX and BCL2, association of these two proteins and mobilization through the membrane. But we're not going to talk about that today. But basically, all these extrinsic and intrinsic apoptotic processes convert into the disruption of the mitochondrial membrane potential, calcium release, and also cytochrome C released into the cytosol. Then the cytochrome C release now interacts with the APAF1 component and the caspase 9, and they form this apoptosome. Now the apoptosome can release and so can cleave procaspase 9 and activate procaspase 9. And this goes and activates caspase 3 by cleaving the sites within the procaspase 3 protein. Then caspase 3, because it's considered an executioner caspase, then it goes and promotes apoptotic subcellular cascades. So in this diagram, you can see that the differences between intrinsic and extrinsic apoptosis are how the mitochondrial disruption starts. So there are different routes of causing mitochondrial disruption and cytochrome C release. And then the two pathways will convert into this kind of like brownish area in which now the two pathways converge into the activation or the formation of the apoptosome, activation of caspase 9 and caspase 3, and subsequent cascade events. So Caspase-3 is, as I mentioned earlier, a biomarker for apoptosis, but cannot distinguish between extrinsic or intrinsic apoptosis. So, depending on whether you are interested in dissecting the two pathways or whether you're just interested in knowing whether there is an apoptotic mechanism going on, then caspase-3 may not be sufficient to answer those questions. So then other biomarkers should be necessary to determine which type of specific apoptotic pathways are happening. So as I was just talking about some of the apoptotic biomarkers, well, there are a lot of them. Um, Actually, I put here a reference about a lot of different biomarkers, especially those that distinguish apoptosis and necrosis. And there is a really long list of biomarkers you could test. The different apoptotic biomarkers can actually be classified as either intracellular or extracellular markers. And some of those have to, the most studied ones or the most specific ones are those related with intracellular biomarkers, specifically caspase enzymes. And there are two types of apoptotic biomarkers widely used in neuroscience, which is caspase-3 cleavage and activation, and also the presence of the caspase-cleaved cytokeratin 18. So whenever you see caspase-cleaved cytokeratin 18 products, then you know for sure that there is an apoptotic cascade occurring. Then they're not present here, but you could potentially look at BCL or BAX levels, PUMA levels, activation of P53 as markers for intrinsic apoptosis, while you could be looking at activation of caspase 8 as a biomarker for extrinsic apoptosis. And in this slide, I wanted to bring back what we've learned in the previous video about necrosis and do a side-by-side comparison with apoptosis, because necrosis and apoptosis are probably the most important cell death mechanisms that participate in neurodegeneration. So in the case of apoptosis, this process can be both a physiological process as part of the regular dying during development and during other type of physiological processes, but also it's a pathological pathway and affects single cells. So it is different from necrosis that is kind of like spreading across different cells. This is apoptosis. It's a more specialized and a specific mechanism that can affect single cells and not the surrounding area. And in this case, the cell is shrinking. So it is an active process. It is kind of like a consuming process, and that's what I wanted to represent with these two pictures, right? In necrosis is more like a bomb exploding, and everything is kind of like destroyed. In the case of apoptosis, it's more like the deterioration, a slow deterioration of a cell. This is considered an active process because it requires the synthesis of new proteins. There are new proteins such as some of the BCL2 pro-apoptotic family members that get transcribed and synthesized, other mitochondrial proteins, some other factors are synthesized de novo as well. And in this case, what happens with the DNA is that there is a chromatin condensation. A bigger difference, or a really big difference between apoptosis and necrosis, is that in apoptosis, the plasma membrane is intact. And sometimes it's very difficult to look at, to see on a preparation whether there is apoptosis happening or not, because cells may not look very different morphologically. While in necrosis, there is this swelling and membrane disruption. And normally there is no inflammatory reaction with apoptotic pathways. So this is a list of like differences between necrosis and apoptosis. And I really encourage you to read this list and really make sure that you understand the biggest differences between these two cell death pathways. And finally, I leave you here with a short quiz to revise the most important concepts about this video. We will see this in class, and we will talk about any other specific or important concepts that have been revised in this video. And finally, I leave you with a new class problem here so you can look at it, and then we will do this problem in class in which we will try to design an experiment to solve whether apoptosis is a possible way of explaining how motor neurons are dying during amyotrophic lateral sclerosis, and how we can combine this study with transcriptomic analysis to determine or analyze specific gene markers for apoptosis and necrosis.